last folder, we finished up talking about kidney anatomy pretty in depth, and we looked at the structure of the nephron, which is responsible for actually filtering the blood and from that producing urine. With our next series of videos, we're going to be talking a little bit about the process through which urine is actually created. So it's a three-step process. The first step is known as filtration, the second is reabsorption, and the third is tubular secretion. With this particular video and this particular folder, we're going to be focused on just the first step of urine formation, which is, again, filtration. And this is something that's happening at the glomerulus. So it's occurring between the glomerulus and the glomerular capsule, which again is that initial segment of the renal tubule. If you look at this picture over here, we've got an artist's very simplified representation of this area. So here's our glomerulus. Here's that glomerular capsule and the rest of the renal tubule that the glomerular capsule is actually attached to. I want you to notice something that isn't exactly portrayed well in this particular diagram, but I want to talk about it a little bit, and that is the afferent arteriole and the efferent arteriole. So the afferent arteriole is the structure of the glomerulus that brings blood down here into the capillary knot where it's actually filtered. The efferent arteriole is the blood vessel that's taking blood away from the glomerulus once it has been filtered. And you'll notice that that efferent arteriole comes down and actually runs along the renal tubule and becomes the peritubular capillary. But there's something that's very specific that's going on with the afferent and efferent arteriole that allows this process of filtration where things are being pushed out of the blood into the renal tubule to occur as a passive process. And what I mean by that is there is no energy in the form of ATP that's having to be used to physically pump substances out of the blood and into the glomerular capsule. What actually happens instead is the efferent arteriole decreases its diameter, so it constricts to some extent, and its diameter is much smaller than the diameter of the afferent arteriole. Now, this is what I was saying is not represented on this diagram well. Really, they look like the afferent arteriole maybe is more constricted, or at best they're equal, and that's not the real life situation that's happening in our nephrons. The real situation is the efferent arteriole is constricted, the afferent arteriole is dilated to some extent, and what that means is the influx of blood into the glomerulus is not matched by the efflux of blood. Basically, we've got more blood and things that are contained within the blood coming into the glomerulus than we have leaving. And by having this constricted blood vessel here, what it does is it creates an area of high pressure in the glomerulus, and it's simply that pressure that's found within this blood vessel that tends to push substances in the blood through the filtration slits and the fenestrated capillaries that we have here, and also through the fenestrations in the blood vessel itself. So filtration is a passive process. We're just creating an area of high pressure in the glomerulus, and that's pushing substances out of the blood and into the renal tubule. The other thing that I want to say about filtration, just generally to kind of get you familiar with it, is virtually everything that's in the blood. So fluids like water, hormones, ions, nutrients like glucose, vitamins, all of these things are small enough to be pushed out of the fenestrations in the blood vessel wall and also pushed out of the filtration slits, and they make their way into the glomerular capsule. That means that virtually everything that enters the glomerulus, with the exception of blood cells, so red blood cells, white blood cells, which are large, and also some very large proteins that are found within the blood, virtually everything else gets pushed out of the blood and makes its way into the renal tubule. So that's some general stuff about filtration. What I want to do next is talk about some specifics and some mathematical relationships that are controlling how much actually gets pushed out of the glomerulus during the process of filtration. Before we get into that, there's one last thing that I want to say. It's a term that you should be familiar with, and that term is filtrate. And the filtrate is basically everything that's being pushed out of the blood and into the renal tubule. So if it came out of the blood and it's in the renal tubule, we refer to that as the filtrate. So what I want to do with this video is talk about a term that's really important that you'll definitely want to be familiar with, and that term is net filtration pressure, or NFP. NFP is the pressure in the glomerulus that's driving fluids and nutrients and glucose and vitamins and all those things that I talked about out of the blood and into the glomerular capsule. So this is the pressure that's causing filtration to occur. And I've talked about already, and this is represented much better on this diagram, the fact that we've got this constricted efferent arteriole and this dilated afferent arteriole, which creates an area of high pressure here in the glomerulus. So blood pressure is just kind of relatively high in the glomerulus. It's actually equal to about 55 millimeters of mercury. And this is the force that is pushing things, fluids and other substances, out of the glomerulus and into the glomerular capsule. It's sometimes also referred to as the glomerular hydrostatic pressure, or the HPG. So when we're talking about a hydrostatic pressure, we're talking about a pressure in a fluid, and effectively this is just the blood pressure, blood's mostly a fluid, right, within the glomerulus itself. Something that's a little bit maybe less intuitive than this is that there are actually forces that are opposing the glomerular hydrostatic pressure. So if you look at this diagram over here a little bit more closely, we've got these purple arrows that are indicating that blood pressure that's pushing stuff out of the glomerulus. But you'll notice we've got some red arrows that are a force that's pushing against that, and some green arrows that are doing the same thing. What I'm going to do for a minute is talk about these forces that are pulling fluid, if you will, back into the glomerulus, or pushing it out of the glomerular capsule and back into the glomerulus, because there are a couple of forces that are doing that. So the first of these is the capsular hydrostatic pressure, or what's also known as the HPC. We've talked about this idea that blood is pushing substances out of itself into this capsule. And so we're getting a lot of fluids and small proteins and other things building up in this capsule. And those things collectively are creating a fluid pressure that's a back pressure on the glomerulus that's tending to oppose substances moving out of the glomerulus. And that's our capsular hydrostatic pressure, or HPC. That particular pressure is equal to about 15 millimeters of mercury. And it's equal to, or it's represented by, the green arrows on this diagram. The second force that's tending to pull things back into the glomerulus, or push things, if you will, from the glomerular capsule back into the blood, is what's known as the colloid osmotic pressure of glomerular blood. And I want to draw for you a diagram to kind of represent what this is, because it's maybe a little bit more complicated. So here's my very, very simplified representation of the actual glomerulus. So the blood vessel through which blood is going to move and be filtered. I'm going to change the color to hopefully make this a little bit more easy to differentiate. And I'm going to then draw in the glomerular capsule. So I'm drawing a picture very similar to the one that you already see here, so that we can represent what's going on with this colloid osmotic pressure. Now you guys know at this point that blood consists of blood cells and proteins, and then there's a lot of other things, and these other things are small, and they're things again like water and small proteins and nutrients and those kinds of things. So I'm going to draw in, I'm going to use green just so we've got some color differences, these blood cells and these large proteins. So these little green circles are going to represent those in the blood. And I'm going to use blue to represent everything else that's smaller. So this would be your water, your nutrients, those other things that make up the bulk of the blood. What we see happening as blood enters into the glomerulus is it's got a lot of these small substances, water and such, and a lot of these blood cells as well. But what happens over time through the glomerulus, as blood is traveling through it, is these small substances are pushed out of the glomerulus and into the glomerular capsule. So they start to move into this glomerular capsule just because they're small enough to do so. And the blood cells and the large proteins are not. So they stay on this side of the membrane. They stay within the glomerulus itself, and their concentration starts to increase as the blood is continuing to move through the glomerulus, because they're simply not able to move. What this does is it sets up a concentration gradient. So now, by the time we get over here, we have this membrane, and on one side of the membrane, we've got lots of water and all these other small things. And on the other side of the membrane, we have these large proteins and these blood cells. And what's happening now is that we have a concentration gradient, whereby our water is all over here for the most part, and our large proteins are all over here. Our large proteins simply can't move into the glomerulus, or into the glomerular capsule. They're not able to do that because they're too big. And so what happens is we get an osmotic pressure developing, and that osmotic pressure tends to pull stuff back into the glomerulus from the glomerular capsule to try and establish equilibrium once again. This is what the blood colloid osmotic pressure is, and it's equal to 30 millimeters of mercury. So if we were to calculate the net filtration pressure, again, this is the overall pressure that's pushing stuff out of the glomerulus and into the glomerular capsule, we have to account for both the force that's pushing stuff out, but these two forces that are tending to pull stuff back in as well. We've talked about the force that's pushing stuff out and the fact that it is equal to 55 millimeters of mercury. We've also talked about the capsular hydrostatic pressure being equal to 15, our colloid osmotic pressure, which is the one that we just finished up talking about, equal to 30. So these two together are equal to 45. And basically what we get for a net overall pressure is, if things are healthy in the kidneys, we're maintaining the blood pressure where it should be, the pressure pushing stuff out is equal to 55 millimeters of mercury. The collective pressures pushing stuff back in is equal to 45 millimeters of mercury, and we get an overall net filtration pressure of 10. So the overall pressure, when we account for all these different forces at work at the glomerulus, is equal to 10, and that's the pressure that's driving substances out of the blood. And the body tries to regulate that pretty highly to keep that pressure consistent over time as long as things are normal with blood pressure. Here's another term that you definitely want to be familiar with. It's really important, and you're going to want to understand how it's related to the net filtration pressure. And this is what's known as the glomerular filtration rate, or the GFR. So here's a definition of what GFR is. It's the volume of filtrate that's being formed. So remember, filtrate is everything that gets pushed out of the blood into the renal tubule. So the volume of filtrate that's being formed each minute by all the nephrons in the kidneys working together. This averages about 125 milliliters a minute for adults. And typically what we see is the kidneys try to regulate the GFR and kind of keep it within that area. We don't want GFR getting really high under most circumstances, and we don't want it getting really low under most circumstances. We want the kidneys kind of chugging along, producing filtrate at this rate of 125 milliliters a minute. However, there are situations where we may want to increase GFR and where we may want to decrease GFR. And so I want to talk about some of the ways that the kidneys can actually go about doing this. So I've got a list here of 3 things that are capable of either increasing the GFR when needed or decreasing the GFR when needed. So the things that affect this are the total surface area available for filtration, the filtration membrane permeability, and the NFP. And I want to use this diagram over here to talk a little bit about what each of those things are. So if you look at this diagram, we're taking this area of the glomerulus, glomerular capsule. Here's that afferent arteriole, the glomerulus itself, efferent arteriole there. And we've cut it so that you can see kind of the internal structures. So to reorient you, here's our afferent arteriole bringing blood into the glomerulus. These little blood vessels that are cut in cross section are also, or actually, the capillaries of the glomerulus itself. And then we've got the efferent arteriole that's taking blood away. I want you to focus for a minute on these purple cells that are kind of interspersed in between the different branches of this capillary. These are what are known as mesangial cells, and they are capable of contracting. And when they contract, they are going to put pressure on the walls of the glomerulus, and they are going to collapse those walls to some extent. That can decrease the surface area that's available for filtration, because remember, it's across the walls of the capillaries that filtration is occurring. So if these cells are contracting, they can decrease the surface area for filtration. If they are relaxed, they can increase the surface area for filtration. The other thing is, remember, these are fenestrated capillaries. So if we contract their outer walls, that's going to decrease the pore size that's contained within the wall itself. It's going to make those fenestrations smaller, and that's going to decrease the permeability of the filtration membrane. Not as much stuff is going to be able to move through the filtration membrane if those fenestrations are closed up. If the mesangial cells are relaxed and those pores are more open, that's going to allow more stuff to be filtered out, which would increase the GFR to some extent. But the primary thing that's affecting the GFR is the NFP, the net filtration pressure, which, remember, is highly dependent on the blood pressure within the glomerulus. And what we see between the NFP and the GFR is that there is a direct relationship. So when net filtration pressure increases, it makes sense, if there's more pressure in the glomerulus, we're going to push more out into the renal tubule, and we're going to increase that GFR. If we see a decrease in NFP, which would likely be due to a decrease in blood pressure specifically within the glomerulus, there's less pressure pushing stuff out in those situations, and we're going to then see a decrease in GFR. Less filtrate's going to be formed simply because there's not as much pressure pushing stuff out. So I mentioned previously that the rate at which filtrate is formed, so the glomerular filtration rate, or GFR, is something that the body tries to keep consistent. So it's about 125 milliliters a minute. And under normal, healthy circumstances, blood pressure is good in the body. The kidneys work really hard to keep it just at that through what are known as intrinsic controls or renal autoregulation. So auto means self. Renal autoregulation is the kidneys basically regulating themselves and working to ensure that they are able to keep this glomerular filtration rate within the range that it needs to be. If you have a really severe drop in blood pressure or a really severe increase in blood pressure, it no longer makes sense to continue producing filtrate at the same rate. If your blood pressure is really high, one of the best things that we can do to bring it back down or prevent it from getting higher is to get rid of blood volume. So if we increase the filtration rate, if we increase the amount of filtrate that's being formed, we're going to be taking a lot of fluid volume from the blood, and that can help to decrease blood pressure. So under a situation where at high blood pressure, you may see GFR increasing as we try to get rid of excess fluid in the blood. Conversely, if blood pressure is really low, one of the best ways that we can prevent blood pressure from getting any lower is to maintain fluid volume in the blood. So it doesn't make sense, if you've had a severe decrease in blood pressure, to continue chugging along, filtrating 125 milliliters a minute of fluids and other substances out of the blood. It instead makes sense to conserve those fluids in the blood, to keep them in the blood, to prevent a further drop in blood pressure. So under that situation, again, where blood pressure is abnormally low, we would see a situation where GFR would be decreased. When this happens, when you've got either a big increase in blood pressure or a big decrease in blood pressure, glomerular filtration becomes under the control of what are known as extrinsic controls. So they are extrinsic controls because they are outside of the kidneys. They're extrinsic to the kidneys. Actually, what we see happening in situations where blood pressure becomes too high or too low is that the nervous system and the endocrine system get involved, and they either up the amount of filtrate that's being produced to help with blood pressure if it's too high, or they decrease the amount of filtrate that's being produced to help with blood pressure if it's too low. But these extrinsic controls are going to maintain blood pressure and not worry about keeping the filtration rate where it normally is if there's a blood pressure problem in the body.